Okay, now we begin part two, and part two starts with a discussion of microtubules, the largest of the cytoskeletal components. And what I'm going to basically do here is compare and contrast microtubules to actin and tell you mostly the contrasts, what's different about microtubules. And I'll just kind of focus my discussion on those differences. So much of what we learned for actin filaments can be applied to microtubules. We have similar concepts. Monomers link together to form polymers. Note that that linkage is not a covalent attachment. So we're not making a covalent bond when we link these monomers up together. We're using instead weak force attractions to hold the monomers together into the polymer. And weak force attractions, of course, include things like van der Waals interactions, hydrogen bonding, hydrophobic interactions, ionic attractions, those sorts of non covalent uh, mechanisms. So here, the nature of the monomer is a little bit different. So the monomer for microtubules is a protein called tubulin. Tubulin is what we call a heterodimer. Hetero for different, dimer for two. So it's two different subunits interacting to form the monomer. One of the subunits is alpha tubulin, the other is beta tubulin. And alpha tubulin and beta tubulin, that the artist has shown as like light green and blue here, physically interact through weak force attraction to form the tubulin monomer. Each of these is a unique protein product encoded by a unique gene. So an alpha tubulin gene gets expressed, we get the alpha tubulin protein. A beta tubulin gene gets expressed, we then get the beta tubulin protein. And then they interact together to form a monomer of tubulin. So tubulin is a heterodimer of alpha and beta isoforms. Now we get nucleation. So we get the formation of a nucleus, just like we saw before. And once we reach the critical concentration, we start to build the filament. And just as we saw for actin filaments, microtubules have a polarity to them. So they have a plus end of the filament and a minus end of the filament. And there's preferential addition to the plus end, just like we saw before. We also have a similar sort of graph that we looked at before, where once we get the critical concentration of the heterodimer, the monomer, we start to build the filament very, very quickly. Just like actin filaments, microtubules are quite dynamic, and their filamentation can be either stabilized or destabilized, depending on interactions with other proteins. So all of those kind of dynamics are the same. And the other thing to remember, and the micrograph here gives us a little bit of a look at that, is the way the microtubules are arranged. They're sort of like train tracks. And if we draw kind of a general cell with a nucleus, eukaryotic cell obviously, the train tracks kind of radiate out like this. And the way I think about this from a metaphor perspective is around the nucleus, this sort of perinuclear area, there's a microtubule organizing complex where all of the minus ends of the filaments are sort of anchored. Think of this as like the, Grand Central Station in New York, and all of these train tracks radiating out are the feeder lines that come in to the station. And so it's sort of analogous to that if we're going with the train metaphor. The plus end of the microtubules are the ones found towards the periphery of the cell. Okay, so the polarity of the filament is such that the minus end is towards the nucleus, the plus end is towards the periphery. And what these train tracks are doing, just as the train track system here in the US is delivering, cargo from one place to another, same thing with the microtubules, except now the cargo, instead of being, cars or coal or raw materials, whatever, is protein cargo or organelles. And that's how they sort of move through the cell. The other thing to remember is that with actin, we noticed that an actin monomer is more stable in the ATP bound state. Something similar is true for tubulin, but instead of ATP, it's GTP, another trinucleotide phosphate, but guanine instead of adenine. And so this one is GTP bound in the more stable state. So here's a closer look at it, and this is sort of similar to what we saw with actin. The GTP bound monomers are the ones that are preferentially added to the plus end of the filament, and tubulin, just like actin, has an intrinsic ability to hydrolyze the bound trinucleotide. So when that hydrolysis kicks in, you see the inorganic phosphate leaving. We're left with GDP-bound monomers towards the minus end of the microtubule. And those are more prone to disassembly, just as we saw with actin. And the other thing that's similar to actin is the fact that microtubules have binding proteins as well. They're called MAPs, microtubule-associated proteins. And what these MAPs do is they help to affect the dynamics. Some of the MAPs are stabilizing. They help to stabilize the filament by toning down the GTPase ability of tubulin. Others speed up the rate of the GTPase activity and help to destabilize the filament. So very much the same dynamics as we saw. Treadmilling occurs with microtubules just as it does with actin. And capping can occur as well to stabilize the polymer. So another kind of cool thing about microtubules is that they are clinically relevant. Quite a few of our cancer treatment drugs target microtubule stability. And the logic here is a tumor, a cancer cell that's become transformed, is rapidly dividing, has a very high metabolic rate. And so if we could destabilize microtubule polymers when the tumor cell tries to divide, we might be able to disrupt or slow down the rate of its cell division. Cancer is essentially uncontrolled cell division. If we could slow that down, it would be helpful. So many of the therapeutic drugs that we use for cancer treatment are able to alter microtubule dynamics and to speed up their dissociation. Taxol and colchicine are two of those drugs that help to alter microtubule dynamics by encouraging destabilization instead of stabilization. They work through different mechanisms, and I'm not really going to talk about those mechanisms, but they are important from a clinical relevance perspective. Okay, so if the microtubules are the train tracks and vesicles loaded with protein are the cargo, what are the trains that form, what are the cars that form the actual train that move the cargo? I know the metaphor is a little strained, but bear with me. So the train is made up of these various molecular motors. I'm going to circle one of them here. This is called kinesin. And attached to kinesin would be a vesicle containing the cargo. So this is kind of like the car on the train that holds the cargo. And then that what are called head domains of the kinesin complex are physically bound to the outside of the microtubule. And this head domain kind of walks systematically along the microtubule. And the way that that works is the head domain can bind to ATP and then hydrolyze that ATP, and the energy from that hydrolysis changes the shape of the head domain and moves it one subunit forward. So a whole lot of ATP hydrolysis, a whole lot of ATP is consumed in making these motor proteins work. Kinesin is the motor protein that moves cargo towards the plus end of the microtubule, so away from the nucleus, towards the periphery. Dynein is the molecular motor that moves from the plus towards the minus end, or from the periphery towards the nucleus. So when we talked about the endomembrane system and we talked about material moving through the cell, one thing I didn't tell you is that the transport vesicles that move the material from one place to another have to get hooked up to a motor protein to move. So when a vesicle comes out of the Golgi, for example, ready to move up to the cell surface for secretion, it's not magical diffusion, stochastic thing that helps to regulate it. It's very regulated. The vesicle will get hooked up to kinesin, and kinesin will hook up to the microtubule and then move the protein up towards the periphery. Similarly, for the degradation side of the endomembrane system, when endocytosis occurs and we deliver a clathrin-coated vesicle to the endosome, that movement from the plasma membrane to the organelle is happening by connecting that vesicle to dynein. Okay, it's not just sort of magical diffusion that's doing all of that. So these molecular motors are critical, and the use of the microtubule train track system is also critical. A couple things to note before I move on. A common misconception, because the microtubule is hollow, if we looked at the head of it, we would see the tubulin heterodimers are kind of arranged, and there's a lumen, but nothing travels in that lumen. All the traveling happens by connection to the outside of the microtubule. So don't think that things are being extruded through the lumen of the microtubule. That simply isn't the case. It's just the way that the polymer is built. It happens to build a hollow polymer instead of a tight polymer. So now we're going to look at some experimental approaches to study the role of microtubules and the motor proteins in sort of moving protein cargo through the cell. And historically, the model system of choice has been a neuron in which to sort of examine these dynamics. The reason the neuron is a really useful model is they're very big cells, highly polarized. You know, here's the soma, here's the axon. The axon is made up of, there's a bunch of stuff in there, but one of the key things in there are microtubules. The microtubules, those train tracks, are there to deliver cargo that's made in the soma to the end of the axon where it's needed. So for example, a lot of the protein components of the end of the axon are things like voltage-gated ion channels that help to turn the action potential into, chemical energy that can cause the neurotransmitters to be released. A lot of the neurotransmitter synthesizing proteins work down here at the end of the axon. But where are those proteins made? There's no ER, there's no secretory pathway down here at the end of the axon. All that stuff's up here in the soma. So the proteins that work way down here have to be synthesized up here and then transported down the axon. So the neuron and its structure provides a really useful model system in which to take a look at microtubules, motor proteins, and how they move cargo through the cell. And the typical way that we study this is to do something akin to a pulse chase experiment in vivo. So let's imagine that, this is a cartoon representation of the spinal cord. The spinal cord runs from the head of the animal to the tail of the animal, if we're talking about a mouse, which is where most of this work is done. And lined up along the side of the spinal cord are these collections of soma. And these collections of soma are called the DRG. So over here, I'm going to try to circle the DRG. This is a collection of soma, so it's analogous to this. And connected to the soma is this really long axon that makes up a nerve. It's called the sciatic nerve. It doesn't matter what the name of that nerve is, but it's written there, so I'll mention it. So the soma's here, in the DRG, and then the axons travel in the sciatic nerve to the target, the skin or the muscle in the periphery. So we can inject radio-labeled amino acids into the soma. And then in the soma, protein synthesis will occur in the presence of those radioactive amino acids. And we'll get a pool of radioactive protein that's produced. And so those proteins will be packaged into vesicles. The vesicles will be hooked up to kinesin, and the kinesin will hook up to the microtubule and begin transporting that material down the axon. So we can harvest or collect the axons at different points in time after the injection of the radioactive amino acids. So let's imagine that we can collect the axon and then we can segment it. We can dissect it and make these different segments. And I'll label them segment 1, 2, 3, and 4. And then from each of these segments, I can extract protein that I can then load into a gel. So the protein from segment 1 gets loaded into this first lane of the gel. The protein from segment 2 gets loaded into this lane, and so on. And we do this in multiple animals and at different times. Time 1 is just a few minutes into the experiment. Time 2 is later, time 3 still later, time 4 latest. And take a look at what we see. At the earliest time point, for the segments that are further away from the soma, there hasn't been enough time yet for any protein to be transported into that segment. But for the segments 1 and 2 that are closer to the soma, we're starting to see these proteins that are moving. And for segment 2, we see this kind of red pool of protein that's moving faster than this blue pool of protein. The red pool of protein has made it a little bit further down the axon than the blue pool at time 1. Time 2 is a period of time later. And now we see that the red pool that was migrating faster here has made it all the way to segment 4. Our blue pool has made it into segment 2. And now we have a new purple pool that's moving slower that's only made it into segment 1. And you can just kind of follow that pool through. By time 3, the purple pool is in segment 2, and now there's a new darker purple pool, and so on. I think you get the idea. So what this tells us is that proteins move at different rates, right? And that they're moving from their site of production, the soma, into the axon to their site of use at the end of the axon. So again, the neuron, a pretty dramatic example of microtubules and motor proteins in action. If we were to treat this mouse with a drug that would destabilize the microtubule network, of course it would disrupt all of this transport, for example. So that would be one experimental manipulation we could imagine. So why did I go through all of that, and why is it important? Well, the reason it's important is it helps us to understand how cargo moves along the microtubules. And the other reason that I'm spending time talking about this is, believe it or not, transport of cargo along microtubules is related to our research lab. And let me walk you through the logic of that. So I told you in my lab lecture last week that there's this protein APP. APP includes a sequence of amino acids known as the A-beta domain, or the A-beta sequence. And we talked about how proteases can cut in 2 places to liberate the A-beta fragment. That can get secreted and cause plaque and cause Alzheimer's disease. The secretase enzymes involved are beta secretase that cuts here and gamma secretase that cuts here. I didn't tell you the more formal name of those enzymes. I'll do that now. The beta secretase is known as BASE, and the gamma secretase is known as presenilin. So remember that if we think about studying APP and its role in Alzheimer's disease, there are a couple ways that we can do that. One would be to make a mouse model where there is human APP expressed on top of the normal APP in the mouse to give extra APP. And if we did that, we would hypothesize that it might give us elevated amyloid beta and plaque and a good model of the disease. That's called a transgenic approach. Make a transgenic mouse that overexpresses human APP to give us a mouse model of Alzheimer's. That's been done and it's been very helpful. The other thing we could do to study the role of APP is to knock out the gene that encodes APP. So now in the mouse, take the APP gene and eliminate it. Make a knockout mouse. And then we can infer function by looking at the difference in the knockout mouse from a phenotype perspective compared to the wild type mouse. And any difference that we would see would logically be because of the elimination of the APP gene. And that's a simplistic way of stating it, but that is essentially the essence of the knockout approach. Let's get rid of the gene, see what gets screwed up, and then infer function based on what the screw up is. It's a classic approach. So this was done in the year 2001 and published in Nature. So some scientists decided to knock out APP in the mouse to take a look at its function. And one of the surprising observations was the mouse didn't transport cargo along the axon as well as the wild-type mouse did. That suggested that there was some role for APP in the transport of cargo along microtubules. Up until then, we had no idea what the normal role for APP was. We knew that when it's proteolyzed abnormally, we get extra amyloid beta and it causes Alzheimer's disease, but it couldn't have evolved for that purpose. So we were very curious as a group of scientists, what is it that APP is doing? So a couple of really clever people made the knockout mouse, and then we were able to characterize the deficit. Turns out APP plays a role in some forms of vesicular transport. The cartoon here helps us to sort of get a visual of what that might look like. And I'll give you kind of the bottom line first, and then in the next slide I'll show you some data that allowed us to make that conclusion. So we're looking here at a cartoon schematic of a neuron. Here's the nucleus, here's the soma, here's the axon. We're not seeing the end of the axon, it's way down there. And we're kind of zoomed in, and it's not to scale, I don't want this to be confusing. We're showing a single microtubule polymer with its plus end here. And then here's the vesicle with the cargo of interest. Here's the motor protein, kinesin, and here is APP. And what you can see is APP is in the vesicle membrane, and it's linking the vesicle to the motor protein. And that seems to be what APP is doing, at least one of its roles in the nervous system. Please note this is full-length APP. Here's the A-beta sequence. So it's not proteolyzed APP, okay? It's the full-length APP that's doing this. Now I mentioned that the drawing's not to scale. In reality, all of this would be much smaller and would fit inside the cell. Okay, we're kind of drawing it as if it's outside the cell, and that's obviously not accurate. That's just so that we can see it. And here you see presenilin and base as some of the cargo carried within this vesicle. There'd be other stuff in here too, okay? Lots of other proteins inside that vesicle. So that's the bottom line, that's the conclusion. Let's see how this works. So the way that we would examine the role of APP in axonal transport is to compare the knockout to the wild type. So the scientist here did an experiment very similar to the cartoon that I showed you 2 slides ago, where they injected radioactive amino acids into the soma of these neurons, allowed time for the proteins to be made and to begin to be transported down the axon, and then dissected the cells collecting both the soma and the axon, extracted proteins, ran gels, and then visualized protein products. So here we see a couple different blots are shown. One is protein extracted from soma, from the DRG, which is, remember, a collection of soma. The other, protein extracted from the axons, the sciatic nerve. And in each of these gels and the resultant blots, we're comparing a wild type lane to the knockout lane. Remember, it's the APP knockout that's been done. And over here for the sciatic nerve, this is the wild type lane, this is the knockout lane. And then what we're really looking at are a whole bunch of separate blots looking at these particular proteins. And then we've kind of photoshopped it all together into one image. Not we, but the scientists that did this photoshopped it all together into an image that we could see. So this particular part of the gel that I'm outlining is visualization of APP itself. Remember, that's the gene that we knocked out. So we should not see any signal in the lanes from the knockout, and we don't. You see here, we don't see any signal. Here, we don't see any signal. Whereas for the wild type, where we haven't done the manipulation, we should see a nice thick band. Remember that the thickness of the band and the intensity of the band is directly related to the amount of protein present. So if I have a thick band like this in a gel and a blot, that means a lot of protein's there. If I have a thin band like that, it means only a little bit's there. And if I have none, like here, that means that there's no protein present. So let's take a look at some of the other products that they examined here. They looked at BACE and PS1, presenilin 1, but they looked at all these other proteins too. All the ones that I just outlined here, from BACE all the way down to TREC-A, these are proteins that are known to be carried by kinesin, and they're known to be carried by kinesin 1. Kinesin 1 is one of the different versions of kinesin that we see. There's also kinesin 2. And each of these carries different cargo. All of the ones that I indicated here are carried by kinesin 1. So let's take a look at the pattern of data, compare the knockout and the wild type, see what we can learn. Let's first look in the soma of the wild type animal and compare that to what's going on in the nerve of the wild type animal. And I'm just going to choose, we could do any of them. Let's do BACE. So for BACE, that's the cargo protein, there's a tiny signal in the wild type soma. And there's a much bigger signal in the wild type nerve. That's because everything's working normally. BACE is synthesized in the soma, quickly packaged into vesicles, linked up to kinesin, and then transported into the axon. So it's moving quickly from the site of synthesis into the axon. And that's why we see more of the protein accumulating in the axon for the wild type animal. Compare that to the pattern that we see for the knockout. We see a much bigger band in the soma and a much lighter band in the axon. That means that the protein is being made and it's accumulating in the soma. That's because there's some deficiency in the transport down the axon. And that's why we see less of the protein present in the axon of the knockout compared to the wild type. And note that that same pattern holds for all of the rest of these proteins that are kinesin 1 transported products. So what's happening is the protein's being produced in the soma of the knockout, but it's not moving as well into the axon. And the inference that we would draw is that APP must play some role in the efficient transport of the protein into the axon. Now, of course, what we know is that APP is linking the transport vesicle to the motor protein kinesin 1. What about kinesin 2? They looked at 3 different cargo known to be carried by kinesin 2, and you can see that the pattern of data between the knockout and the wild type is the same. There's no difference. So therefore, APP doesn't seem to be playing a role in the transport of kinesin 2 material. They also looked at tubulin as a control. Tubulin is, remember, the monomer that's used to make the microtubule polymers. And tubulin is also unaffected by APP knockout. But APP knockout does affect a subset of kinesin-mediated transport, and it seems to be all the cargo carried by kinesin 1. Now, a couple things to note here related to these 2 points. You'll see that for the APP knockout, for all of the kinesin 1 cargo, there's still a little bit of a band in the axon. We haven't eliminated transport altogether by knocking out APP. So there must be a redundant protein that can still do APP's job, at least a little bit, not as well, but a little bit, when APP is gone. This is the concept of redundancy, and we see a lot of redundancy built in to eukaryotes, and it comes about because of gene duplication, making different versions of different proteins that then sort of evolve different roles over time. There are APP-like proteins, we call them APP-LPs, that are also able to link transport vesicles to kinesin 1. They just don't do it as well as APP does. So when APP is missing, the APP-LPs can pick up some of the slack, but not all of it. And that's what's giving us a little bit of a band in the axons of the APP knockout. Okay, so we've taken a look in this lecture at the cytoskeleton. We have focused on actin and microtubules in particular, and we've looked at some specific examples related to those microtubules in the functioning of a cell. The other cool thing we've done is we've linked lecture material to what we're doing in the lab, and we're looking here at the role of APP. We learn about the role by making a knockout and then characterizing the knockout compared to the wild type. And we infer from the pattern of data that we see. Okay, so on Wednesday, Dr. Shaven will be posting Lecture 7. Lecture 7 is really dealing with how the cell fuels all of its biochemistry. So how do we get the fuel to do all these things? It focuses a lot on the enzymes that drive metabolism and on the basic metabolic pathways that give us ATP as fuel for the cell. Once that lecture is done, that's really the end of Unit 1, where we've defined the cell and its study. And then Dr. Shaven will be taking over for Unit 2. She'll be doing most of the lectures in Unit 2. I'll be doing one towards the end. She is focused on, for Unit 2, the flow of information through the cell. How does the central dogma work from a mechanistic perspective and a regulation perspective? So she'll be looking at each of the phases of information flow and talking to us about how the molecular mechanisms work. How does replication work? How does transcription work? How does translation work? But more importantly, how these processes are regulated. She'll try to link that to the cell cycle. And then once we get up to the protein level, I'll give you a sense of how post-translational modifications of proteins happen. Okay, so that's enough for today. I hope you all have a wonderful day and a wonderful week 3. And I will talk to you all again on Thursday when we do the pre-lab lecture. Take care.